Because when you get an infection, your body doesn't just need a generic defense.
No.
It needs specific soldiers. The highly trained T cells, the B cells making antibodies, the rapid response NK cells.
And it needs them fast. Manufactured, scaled up, and sent to the exact spot, whether that's a tiny patch of skin or one specific lymph node.
And that precision is really the core of our deep dive today.
Yeah.
Our sources lay out this fascinating two-step masterpiece.
Okay, what are the steps?
First, how the body runs a cellular factory line, custom making these white blood cells on demand.
The manufacturing.
And second, how those cells follow this intricate system of chemical messages. It's a mechanism called directed homing. It's like a biological GPS guiding them to their destination.
So that's our mission today. We're going to unpack this incredible logistics puzzle. We'll look at the production line, and then we'll map out that chemical GPS that makes sure these defenders never, ever miss their target.
It's just a masterclass in biology's optimization, making sure no resource is ever wasted.
Okay, let's unpack this then. Let's start with the supply chain, the factory floor.
The factory floor.
Which is, deep inside our bones, in the bone marrow. This is where the initial manufacturing happens, right? Through cell division.
And the key ingredient there, the starting point, is the hematopoietic stem cell, or HSCs. These are the truly versatile, undifferentiated master factory cells. They're the raw material that can become pretty much any blood component the body needs.
So when you say any, you mean everything in the circulatory system?
Everything. We're talking red blood cells for oxygen, platelets for clotting, and, crucially for our conversation, all the different lines of white blood cells.
And that includes the specialized cytotoxic fighters we mentioned, the T cells, B cells, and NK cells.
That's right. And what's so critical is that the stem cell has to make a decision, a choice. Am I going down the lymphoid path to become a B or T cell, or am I going down the myeloid path for something else?
So what tells it what to do? And maybe more importantly, how many to make? Because if you're healthy, production is just kind of cruising along.
Right.
But if you get hit with the flu, you need a massive surge.
That whole decision process is run by chemical communication. The body literally orders these cells like you'd order from a menu.
Using what, exactly?
Using chemical messengers, growth factors, and these specialized proteins called cytokines. This is how the system scales up production and does it fast.
Can you give us an example of how targeted these chemical orders are? It's not just a general alarm bell, is it?
Not at all. It's incredibly specific. Let's use a non-immune example first just to see how it works.
Yeah.
If your body needs more oxygen carriers, maybe you lost some blood, your kidneys release a hormone called EPO.
EPO, erythropoietin.
Exactly.
Yeah.
And that EPO travels straight to the bone marrow with one precise order: make more red blood cells. That's it.
Super clear. A specific call for a specific product, but for an infection, things are more complicated. We need fighters.
That's when the cytokines come in. And they're just as targeted. Say you have widespread inflammation and infection. The body releases specific interleukins.
Like interleukin-2, IL-2.
IL-2 is a massive one. It's basically a proliferation signal. It screams at existing T cells and NK cells. Divide. Now, we need an army.
So IL-2 is the scale-up button for the troops we already have on the field.
Yes. But what about making new ones? Or getting them ready?
Right, the initial development.
For that, you might see interleukin-7, IL-7. It's critical for the survival and the initial development of both T cells and B cells. And then you've got IL-6, which is huge for inflammation, but also for helping B cells mature into plasma cells.
The ones that actually pump out the antibodies.
The antibody factories themselves. So it's not one signal, it's a cocktail of commands all happening at once. Development with IL-7, proliferation with IL-2, and final maturation with IL-6.
That is just a staggering amount of coordinated production. So we've gone from one stem cell to mass-producing thousands of specialized T cells and B cells.
They're ready for deployment.
But the body is huge. How do these new cytotoxic defenders find one tiny infection in, say, a deep muscle tissue?
And this is where the real logistical genius kicks in. Once they're manufactured, they're released into the bloodstream, but they are absolutely not wandering randomly.
They're following those chemical GPS signals you mentioned.
Precisely.
And this is where it gets so fascinating for me. We're talking about chemical sense, basically. How are the cells sniffing their way there?
They literally are. The chemicals that create these scent trails are called chemokines. And you can think of it as a gradient.
A gradient.
Yeah, the injured tissue is constantly releasing these specific chemokines. It lays down a trail They get stronger and stronger the closer you get to the source.
So the cell just follows the trail from low concentration to high concentration, and that leads it right to the battle.
That's the core mechanism. So, for instance, if you get a bacterial infection in your skin, the local tissue releases a specific chemokine called CXCL8. You might also see it called IL-8.
Okay, CXCL8.
And neutrophils, the first responders, they have receptors that are just exquisitely sensitive to that scent. So they follow that CXCL8 breadcrumb trail right to the site.
But not all missions are about rushing straight to the fight, right? I mean, T cells and B cells often have to go to a staging area first, like a lymph node, to get activated.
Yes, and that's a key difference between immediate response and strategic deployment. A lymph node releases its own unique, constant chemokine signature. In this case, it's CCL19 and CCL21.
So T cells are drawn to that signal instead.
Right. As part of their normal surveillance duty, they're just engineered to travel toward that specific signature.
Wait, why is that so important? Why go to the lymph node first? Why not just kill the pathogen right where they find it?
Well, they will, eventually.
Yeah.
But the lymph node is the central intelligence hub. It's the briefing room.
Okay.
This is where other cells, called antigen-presenting cells, show the T cells exactly what the enemy looks like. It's where clonal expansion happens.
That's where one T cell that recognizes the threat suddenly makes thousands of copies of itself.
An army of clones.
Yeah.
So CCL19 and CCL21 are the signals that say, report to the training ground immediately to become a fully effective cytotoxic warrior.
Which brings us to the lock and key part of this. How does a T cell know to follow CCL19 to the lymph node, while a neutrophil knows to follow CXCL8 to the cut on your finger?
It's all about the receptor system. Every single immune cell has specialized receptor proteins on its surface. These are the locks, and they are built to fit only specific chemokine keys.
So the combination of locks on your surface dictates your mission.
It dictates your mission completely.
So, using our examples, the T cell that's designed for that strategic patrolling, it must have the CCR7 receptor.
Exactly. CCR7 is the lock that perfectly fits the CCL19 and CCL21 keys coming from the lymph node. Once that cell senses the fit, its orders are clear. Go to the nearest lymph node for instruction.
Whereas the neutrophil, it's equipped for immediate damage control.
Precisely. Neutrophils carry CXCR1 and CXCR2 receptors. They are sensitive to those acute infection signals like CXCL8. Their order is simple. Go to the infection now.
This specialization is just. It means every B cell, T cell, and NK cell has its mission programmed right onto its surface from the get-go.
It's phenomenal.
So now we move to the final stage, extravasation. These fighters, the T cells, B cells, NK cells, are all speeding down the bloodstream, the main highway. How do they actually take the exit ramp?
Extravasation is one of the most mechanically elegant processes in biology. The blood vessel wall, the endothelium, it's usually a really tightly sealed barrier.
It has to be.
For these big cells to get out, the vessel wall itself has to respond to the emergency signal and sort of open a door for them.
What actually happens at the molecular level? I remember the sources used an analogy about the vessel becoming sticky.
The stickiness is the key. When there's inflammation nearby, the cells lining the vessel, the endothelial cells, they express new molecules on their surface, mainly things called selectins.
Okay, selectins.
And as the white blood cells rush past in the blood, these selectins act like Velcro. It causes the cells to start rolling and slowing down. They just tumble along the wall, looking for a place to stop.
So they roll, they slow down. What's the mechanism that actually stops them completely? The firm anchor.
That's the job of integrins. These are highly specific adhesion molecules on the surface of the T cells and B cells. The infection signals trigger the vessel wall to put up matching docking points.
Like the other half of the Velcro.
A much stronger half. When a rolling white blood cell finds its specific docking point, the integrin locks on tight. That's called firm adherence. And then finally, the cell literally changes shape and squeezes between the endothelial cells to get out.
That is so much more complex than just opening a door. It's this multi-stage process. Slow down, lock on, and then squeeze out. But the sophistication doesn't even stop there. The sources talk about organ-specific address codes.
Yes.
Meaning a T cell knows, even after being made, if its job is to patrol the gut or the skin or the bone.
This is really the crucial aha moment in modern immunology. The body doesn't just send generalized reinforcements. It literally tags cells with their intended destination.
So it's like a cellular postal service. The cell gets stamped with an address label that guarantees delivery to one specific neighborhood in the body.
That is the perfect way to put it. And these address labels are usually specific types of those integrins we just talked about. So take, for example, T cells that are meant to patrol the gut.
Okay.
They express a very specific tag called alpha-4 beta-7 integrin.
And what does that tag actually do for the cell?
That specific integrin is designed to interact only with receptors on the lining of the gut's blood vessels,
Yeah.
but pretty much nowhere else. It's a guaranteed ticket. It says, you are a gut-patrolling cell. Ignore all other exits until you reach the intestines.
Which prevents a T cell trained to fight a gut parasite from wasting its time patrolling, say, your lung?
Exactly. Or your liver.
Yeah.
And you see the reverse if the threat is on the skin.
So skin-homing T cells get a different tag.
A different address label. It's called CLA, or cutaneous lymphocyte antigen.
Uh,
And CLA directs them specifically to the blood vessels that feed the skin. If you have an infection involving bone, those cells are guided by another chemokine, CXCL12, that tells them to home in on the bone marrow.
so the system is layered. First, the cytokines order the production of the cells.
Right, the T cells, B cells, NK cells.
Second, chemokines guide them to a general area like a lymph node.
The surveillance zone.
And third, these specialized integrins give them a final organ-specific street address.
That is the perfect synthesis. This entire system is so much more than just general immunity. It's strategic manufacturing, directed deployment via chemical scent trails, specific receptor keys, and finally, a custom address label for high-precision exit.
The efficiency is just breathtaking. We've seen the production line fueled by growth factors like IL-2 and IL-7. We've covered the chemical GPS, the chemokines like CXCL8, and we've looked at the custom address labels like alpha-4 beta-7 for the gut.
And what's so fascinating is that this is all a continuous dynamic conversation. The environment, the bacteria, the virus, it's constantly shouting these chemical signals. And the immune system is instantly processing them and adjusting its strategy in real time.
It's that dynamic communication that lets us survive.
It's everything.
So what does this all mean for you listening right now? It means the speed and precision of your immune response aren't an accident. They are the result of this extraordinary chemical symphony unfolding inside you at this very moment.
Trillions of decisions made by trillions of cells.
Which leads to a final thought for you to consider.
Yeah.
Given the sheer number of distinct chemical signals, the growth factors, the dozens of chemokines, all the different receptors that have to align perfectly for just one cytotoxic T cell to get from the bone marrow to a cut on your skin.
Just think about the scale of that logistical management when your body is fighting a complex systemic illness. When multiple organs are all signaling for different types of T cells, B cells, and NK cells at the same time, the coordination required is, it's arguably the greatest feat of engineering nature has ever produced.